Hi, my name is Ronan da Silva. I'm a postdoc at the Trenholm lab, and today I'm going to present my project, which is about how visual circuits mediate object recognition in the mouse brain. Now, to illustrate the problem I'm studying, I'm going to show this video, which is an algorithm, a computer algorithm, that is able to recognize objects in a visual scene. So you can see it's assigning labels to different objects in the scene. You can see it's labeling cars, a person riding a bicycle with a backpack, people on the street. Um, and that's pretty cool. However, you can also see it's making a lot of mistakes. For example, here on the right, it is assigning the label bus to the front of the store, which is clearly a mistake our brains would never make. And it's also not labeling a lot of things such as buildings and light posts and the satellite antennas, which are things our brains are able to do very quickly. Also, for a computer program to do this is a very computationally heavy process, whereas our brains are able to do this in a split second using machinery developed a few million years ago. So how is it that the brain processes visual information to achieve such a complex feat? We know that the visual brain is made of hierarchically organized areas, which each one encode a type of information that gets combined in the next area to become a more complex representation. For example, you have view 1, which has a lot of cells that like edges and lines. The information from view 1 is sent to view 2 and then combined into simple shapes. Information from view 2 is then combined in view 4 into more complex objects and so on and so forth until the information reaches high associative areas of the brain, such as the inferior temporal lobe, where you find cells in primates that are able to encode for faces of specific people. Now, the primate visual system is very complicated. There are many visual areas that are interconnected in a very complex pattern. So it's very difficult as a model to study how this processing of information happens in the single cell level. On the other hand, we have rodents, which are animals that in the recent years have been shown to have many complex visually guided behaviors and who dispose of very advanced genetic and physiological tools that allows us to examine in much detail the anatomy, connectivity, and function of neural circuits. In comparison to primates, rodents have a somewhat simpler visual system, but that keeps a good level of complexity. There is also a lot of evidence pointing that it is hierarchically organized in a different pattern from primates, but still. And a few evidence indicate that it does have the two streams, or the two information processing streams that we see in primates, the dorsal stream, which encodes for location and movement of things in the visual scene, and the ventral pathway, which encodes for objects and assigns labels to them. So which areas of the mouse visual cortex exactly are important for object recognition, and how do they work? So the first thing we do is to map out the visual system to identify the different areas of the mouse visual system. And then I visit each one of those areas and I run a set of stimulus that it's just a series of images picked from the internet, and see which neurons respond to each image. I make a recording like this. I extract the fluorescent signal from individual neurons using an automated pipeline, and then I have the data of which neuron responded to which image. However, this alone doesn't tell me what is the image that will optimally stimulate a neuron. It just shows which of my images were the best at driving a specific neuron. So here enter our collaborators from Blake Richards' lab in Namila. So here what we're doing is that they're taking my data of neural responses to many, many images, and they're going to create an artificial model, so an artificial neural network that is able to predict the biological neurons' responses. With this, then we can identify an image that will optimally stimulate the artificial neuron and see if that image is going to also drive strongly the biological neuron inside the mouse's head. And by doing this and training a model for each different area of the visual system, then we hope to be able to identify what each area is doing and hopefully find an area that is able to encode for complex objects. And then in the future, once we identify what's more or less what type of image each area is interested in, then we will do a technique called single cell initiated circuit tracing to label a cell that will find to encode a particular feature of interest, and we'll label all its presynaptic inputs and see what kind of information those cells are encoding. And this hopefully will allow us to understand how is information combined by a single cell or by this ensemble of neurons into encoding complex visual information such as objects in the mouse brain. Thank you.